Hello, I'm Dr. Fossel. As you know, I chair the conference, but I'm going to take an odd perspective on this conference. I'm going to, instead of talking about biomarkers per se, I'm going to talk about a different way of looking at how we use clinical biomarkers, one that many of us see as potentially offering an optimal point of clinical intervention. In short, when it comes to biomarkers, I'd like to see if we can not only measure clinical efficacy, but actually drive clinical efficacy. About 6 years ago, some friends of mine, Zavin Kachaturian, Era Kachaturian, and Dick Mose, the Kachaturians are the editors of Alzheimer's and Dementia, and Dick Mose is actually on our scientific advisory board, put out an editorial in Alzheimer's and Dementia. And what they were saying was that we really need a more useful model to explain how Alzheimer's and other dementias work. And they offered 10 questions that any useful model needed to answer, and they're listed here. It not only needs to describe the role that aging plays in this, because most of us ignore this, and I'll come back to that, but it also needs to be able to explain how risk factors play a role in creating the dementias, the clinical progression and the temporal aspects of that progression, the anatomical aspects of it, the histology, how the elements of pathology interact, the ability to explain mixed dementias, the heterogeneity of those dementias, which I'll come back to, and the relationships between the various biomarkers we use, for example, beta amyloid, tau tangles. But it also should suggest a novel and innovative and effective point of intervention. Well, I confess I did not read Zavin and Era's article. Sorry about that. But about 3 years ago, a number of us got together and we were directing an Alzheimer's Association conference on animal models of Alzheimer's. And the question we were trying to respond to was posed really by that first remark, which is that everything works in mice, but nothing works in humans. That's the pessimistic approach that many of us take in research. But then there's the corresponding one investors take, which is Alzheimer's is the graveyard of companies. And this is distressing. So at the end of that conference on animal models, we actually published the paper that you see above there with hyperlink and the DOI reference. But what I was responsible for was summing up the first day. And what I said was that we have a lot of technical approaches, for example, monoclonal antibodies, and targets, for example, beta amyloid. But what we lack is a unified systems model. And specifically, what I said was that we need to be able to explain how the upstream risk factors drive, for example, Alzheimer's, and how they result in the downstream biomarkers we see. We need to be able to understand and explain the intraspecies heterogeneity between and within dementias. That is, why do I get Parkinson's, you get Alzheimer's? And moreover, if we're looking at Alzheimer's, why do I get it at age 40 and you get it at age 90? Why do some of us have executive function problems, others specifically memory problems or verbal problems? Why is there this heterogeneity? Also, interspecies heterogeneity. We know that almost any animal we look at shows cognitive/behavioral detriments with age. The question is, why do they occur in different ways in different species? That is, why do the genes prompt that? And why are the histology different, the physiology different, and the, if I can call it, clinical outcomes different? Also, such a model should be consistent with all the known clinical research data, and it should have predictive validity. It should be able to explain, for example, the monoclonal antibody results we saw with solanezumab or we see with anakinumab, explain why those things happen the way they do. Finally, it should offer a feasible and testable point of intervention. Well, as you can see, those parallel very much the thoughts that Zavin and Era and Dick had 3 years prior to that. And so afterwards, Zavin actually took me aside and asked me if I would write up my model, which I then did. And it came out last year. It promptly generated enormous numbers of reprint requests and invitations to share conferences like this one. But I'd like to get into a little bit of that model itself and explain why it generated so much interest. And really what we're trying to answer is this question. When it comes to age-related dementias, why can't we get any clinical leverage? Why have we been unable to effectively cure them? And when I say that, I'm looking at data like this. This was data taken a week ago off the clinicaltrials.gov website. And what you see is that there were more than 3,000 interventional trials in dementia, more than 2,100 for Alzheimer's, and almost 2,300 for Parkinson's. And by global consensus, essentially none of these have succeeded in altering the disease course of the dementias. This is sort of where we stand, for example, with regard to monoclonal antibodies. This is by no means an exhaustive list, but you're looking at some of the past so-called failures of monoclonal antibodies. The recent one, that anakinumab, that the FDA okayed last month, and then a number that are in the pipeline, including those with expedited status. Again, this is not an exhaustive list, but if you look at even the best of those, in this case, the anakinumab data, you see something like this. It depends on the dose level you're using. But what's interesting is that in all the monoclonal antibody studies we've looked at, sooner or later you find that the downhill course ends up paralleling the placebo vector. And that's really where we stand. There is a temporal step off, but that's not what we want. What we'd like to see optimally is something that actually stabilizes the disease, prevents any further downhill course, and optimally even improves the cognitive function. That's not what we have. It's what we would like to see. So where we are still is this: everything works in mice, but nothing works in humans. And of course, it's not simply mice. It's also, when we look at other species, we find the same problem with translation, whether we look at fruit flies, flatworms, rats, cats, dogs, dolphins, marmosets, vervet monkeys, rhesus monkeys, baboons, or chimpanzees. And I've been bitten by most of those species at one time or another in my professional career. So it's frustrating. But I think the problem is that we tend to be digging in the wrong place. And when you're digging in the wrong place, then it doesn't matter how big your excavator is or how big an NIH grant you have. For example, the anakinumab costs have been in the order of $1 billion. But the outcomes have been somewhat disappointing because I think we're not digging in the right place. The question is, where is the right place? Well, I think that when it comes to Alzheimer's, this is sort of where we stand. We know an awful lot, but we don't understand enough. We have a tremendous amount of data on the physiology, the histology, the clinical findings, but we lack still an overall view of the model. It's sort of like this quote of Einstein's: any fool can know, the point is to understand. And back 500 years ago, the Vatican astronomers knew a great deal about planetary position, but they had no understanding of orbital mechanics. And in some respects, I think that's where we stand. We know a great deal, but we're somehow looking at the data in not a way that's unified and systemic. So what I'd like to do is talk about age-related dementias and using a unified and systems approach to what we know. And when I say unified, let me give you a clear idea what we mean. If we look at a lot of the upstream risk factors, genetic risk factors, toxins, microbial infections, head trauma, for example, we know that they play a role in the clinical disease. And there's some underlying processes that we're having a hard time understanding. And we know a great deal about the downstream outcomes: beta amyloid, tau tangle, microglial activation, inflammatory changes, immune problems. We know a great deal about that, but we still lack some overall understanding that unifies these. And the same is true when we look at these clinically. For example, it is true that Alzheimer's dementia is a clinical syndrome or clinical disease, and it usually is cognitive in nature, but many of the patients also have motor problems. If we look at Parkinson's disease, we tend to think of that as a motor problem, but many of those patients have cognitive problems. So there's often a clinical overlap in patients. The borders are a little fuzzy, a little gray, and while there are real clinical syndromes, there's a certain amount of overlap. The same is true when we look at the histology on autopsy. We see many of these patients have vascular dementias. And the question is, how do all these fit together? And the same is true when we take a broader view at the usual alphabetic litany of diseases, whether we're looking at Lewy body disease, Huntington's disease, Friedreich's ataxia, progressive supranuclear palsy, primary progressive aphasia, posterior cortical atrophy, mesial temporal atrophy, all of these things. When we look at them, we see that there are many times they're real syndromes, but when we look at our patients in neurology, we find that some of them have mixed dementias. They have aspects of a couple of these. And then there are some patients that just don't seem to fit in any of these categories very neatly. That is very typical. So we've seen more and more global conferences on atypical or mixed dementias. And I think over the past decade or two, a lot of us have had this impression that there is something going on that's more fundamental. And what we're seeing is many different clinical expressions as a result of starting at a different genetic and epigenetic and behavioral baseline. So we see one patient get Alzheimer's, one Parkinson's, but there's some unity about the underlying process. So we're looking for a unified model, but we're also looking for a systems model. And by systems model, I mean something a little bit more subtle. This is, to take an engineering example, this is an Airbus 380 cockpit. In the upper right, you see a Pearl 15 Rolls-Royce jet engine. And the lower right, you're seeing a Trent 900 turbofan that powers that jet engine and then one of its fan blades. Well, in engineering, I'm told that if that fan blade fails, you don't simply ask what's wrong with the fan blade. You ask what's wrong with the fan blade in the context of the Trent 900 turbofan system, in the context of the Pearl 15 Rolls-Royce jet engine, in the context of the Airbus 380, at 35,000 feet, going hundreds of miles an hour at a high RPM and high temperature. In short, it's a Gestalt problem. It's a problem of how that component fits into the overall dynamic system. And I think that's what we need to do with regard to aging and age-related diseases too. For example, if we look at COVID, these are the signs and symptoms, the components, the biomarkers, if you will, as you will, of COVID, but they're not COVID. COVID is the system itself. It's the complicated interaction between the COVID virus and the human immune system and a number of other organs. So while we do need to measure carefully all of these various biomarkers, again, if you can't measure it, it's not science. On the other hand, if you can't understand what you're measuring, it's not enough of a science to let you cure, for example, COVID. We need to go a little deeper. The same is true when we look at age-related diseases. These are macroscopic biomarkers of aging, but they are not aging. They are outcomes of aging, the things we measure, the things we complain about. In some cases, they are specific age-related diseases like osteoarthritis, but by themselves, they're not aging. And if we look at the microscopic level, we have the same problem. I can come up with literally dozens of different microscopic biomarkers of aging, but those don't define the aging system or process. They simply give us things to measure without a better understanding of it. The same is true when we look at the dementias. Here are a number of biomarkers that I think all of us are aware of. And again, we can list dozens of others. And no matter how carefully we measure them, they're not enough to really understand the systems process, that is the dynamic process that underlies the disease. And the same is true when we look at this genetically. I know of more than 200 gene predilections that have been correlated with outcomes or clinical risk factors for dementias. And yet, just knowing you have two APOE4 alleles doesn't explain why you get Alzheimer's and somebody with two APOE2 alleles does not. Same is true of the other genes. So one of the questions that's useful to ask is one that really Zavin and Era asked in that original editorial, which is, what is the greatest risk factor for age-related dementias? And the answer is aging itself. Well, let me introduce you to a friend of mine. This is Len Hayflick. He just actually got an award this week. And Len is a friend of mine, and it was 60 years ago this year, he first described cell aging in human beings. And this is a quote that he and I came up with when we were chatting about this. And he is right. Almost all of us will agree that when we take an age-related disease like dementia, that aging is the single greatest risk factor, but then we tend to ignore aging per se and go on to other issues. And this is the point that Zavin and Era raised in that editorial 6 years ago and that I raise here. Well, it's one also I've raised intermittently over my 50-year career so far. And some 25 years ago, I gave a talk on this at here, the NIH in Bethesda, as well as 2 published articles in JAMA about this issue. And what I was saying was that if we can carefully understand the fundamental aging process at the cellular, genetic, and epigenetic levels, then there's potential for an effective intervention. The question is, can we do that? So can we intervene at aging at its most fundamental level in age-related diseases like dementias? But that requires that we understand aging at the most fundamental level. And we tend to assume we do, but in fact, we usually don't. So let me introduce you to some friends of mine. Typically, human beings here in the upper right live about 3/4 of a century. Now, that is Coco the gorilla in the upper left. She was a friend of mine. I'm probably the only one you'll ever meet who was wrestled with, bitten, and been bitten by a gorilla, Coco in this case. I was her babysitter for a year out of Stanford. And I'm embarrassed to admit she had a bigger vocabulary than I did in sign language. She died about 2 years ago. And typical gorillas last just short of a half a century. Typical domestic animals, depending on the breed, usually last about 2 decades. And typical laboratory rodents, for example, mice and rats, typically last about 2 or 3 years, again, depending on the breed. And the question is why? Well, clearly you can't simply say it's just wear and tear, it's entropy. You tend to say it has something to do with the genetic differences. So for example, that mouse probably shares about 75% of the protein expressing genes with you and I, and presumably that other 25% and the regulatory genes have something to do with why that mouse ages at about 40 times a faster rate than you and I do. But still, that's not an explanation. We've simply assigned it to a large group of genes. And the same is true when we look at specific species like human beings and look at only one gene. Let me introduce you to some friends of mine again. About 20 years ago, I would gather all the progeric children, this is Hutchinson-Gilbert progeria children, together from all over the world. And these are 30 of them that I took for a tour of the White House. And I took them places you are not allowed to go. And I took pictures you are no longer allowed to take. And we had a delightful time until the 2 children in the front row, Sammy and Ori there on the left row, sat on a pressure-sensitive switch just inside the north portico entrance to the White House that leads up to the presidential quarters. And the Secret Service escorted us out, and we took this picture. Well, these children live an average age of 12.7 years. They die overwhelmingly of age-related diseases. Usually it's strokes, heart attacks, as all of these children in this picture did. So why? Well, we know the gene. It's the lamin A gene. But again, knowing the gene does not tell you how that gene works and why these children age at, again, something like 5 or 6 times as fast as you and I do. Why do they age so quickly? So we know sort of where to look, but that's not an explanation. The same is true when we look at some other interesting outliers. For example, every cell in your body, every somatic cell in your body derives from a fertilized ovum, which the ovum, at least, you got from your mother, who got it from her mother. And that line, the gamete line, the germ cell line, goes back about 4.5 billion years, depending on who's estimating. And during that time, retrospectively, there was no significant age-related damage. And yet, in a matter of decades, there's an inflection, and you and I show somatic cell aging. So why? Well, you can't simply ascribe that to mitochondrial change, because the mitochondria you inherited from your mother also, and that line goes back about 1.7 billion years, depending on who's counting, because there are a lot of various estimates. But again, there was no age-related change in that mitochondrial line when you inherited it from your mother. And yet, if I now measure mitochondrial function, which I'll come back to right now in you, almost all your mitochondria show some subtle changes in their ability to maintain themselves. And in fact, that's the problem. It is not simply a matter of entropy, it's a matter of balanced entropy versus maintenance. And the germ cell line managed to maintain itself almost flawlessly over that time. And the same is not true of somatic cells, for example, in the hand of that 80-year-old man in the wheelchair to the left. If I look at his fibroblasts, keratinocytes, vascular endothelial cells, macrophages, or for that matter, microglial cells and neurons, you'll find subtle defects in maintenance that are associated with aging. In fact, it's sort of like Red Queen theory. This is a quote from Lewis Carroll's Through the Looking Glass, where the Red Queen says you must run as fast as you can just to stay in one place. That's exactly what cells do. But if they don't run fast enough, then you begin to find there are losses in cell function. So for example, if we look at any molecular pool in the body, you'll find that in young, healthy cells, the turnover rate is very fast. In fact, almost all molecular pools get recycled. They're continually being produced, broken down, produced, broken down. And the result is you get very little accrual of damage, signified here by a little red star. But as cells senesce, as cell aging occurs, you find the molecular pools are slower in their recycling rates, and we can measure this. And in older cells, it's such a slow rate that the accrual of damage at equilibrium is fairly high, resulting in dysfunctional molecules and therefore dysfunctional cells. In fact, you can look at this mathematically. This is taken from my Oxford University Press textbook on this. And what this really says is that young cells have a higher rate of turnover per unit time than do older cells. And the result is at equilibrium, they have a lower percentage of accrued damage than do older cells. Well, the outcome of that turns out to be significant, for example, for dementias. So let's take a look at turnover in the central nervous system. If we look at one of our favorites, beta amyloid, but we'll come back to others in a minute, what you find is that young cells in the brain, for example, microglial cells, tend to produce beta amyloid, secrete it, bind it, internalize it, degrade it, produce it. There's a continual turnover of beta amyloid molecules. And the result of that is that denatured molecules tend not to stick around because they're turned over. They're washed out, essentially. Whereas as you get older, you begin to have some of these molecules not being turned over rapidly enough, and you get beta amyloid microaggregates. Now you might ask, what about the roles of genes in this? We know that genes are a risk factor. And in fact, if you are lucky enough to have 2APOE2 alleles, what you find is you make relatively non-sticky beta amyloid molecules. And the result of that is you don't get a lot of accrued damage, and therefore you have few beta amyloid microaggregates. On the other hand, if you're unlucky, you have 2 APOE4 alleles, you make stickier molecules, and as a result of that, your rate of accrued damage is higher, and so you have many more beta amyloid microaggregates, which almost certainly explains why the risk is higher and the age of onset is higher in people with those alleles. Well, regardless of that, as we get older, the rate continues to go slower, and so there's an increasing risk of beta amyloid plaque, again, depending on the gene background. The same is true of explaining the clinical outcomes of these. You get the frank outcomes of neuronal cell dysfunction, synaptic dysfunction, necrosis, and clinical dementia. And it doesn't matter whether you're just looking at beta amyloid or you're looking at tau proteins, mitochondrial proteins, which we'll come back to, inflammatory molecules, and so on. We see these same changes, the same slower rate of turnover, and it's been measured. For example, if I look at mitochondria, about 99% of the aerobic enzymes are imported from the nucleus, and the rate at which they're turned over is slower, and the result of that is more dysfunctional aerobic enzymes. So the ATP to reactive oxygen species ratio goes down with cell aging. The lipid molecules in the membrane are also being turned over, but more slowly with aging, so integrity goes down. Our scavenger molecules, for example, superoxide dismutase, catalase, and so on, go down, so they're less effective. And finally, when there is damage to other molecules, that's also being recycled more slowly. So for at least 4 separate reasons, we see this exponential increase in mitochondrial dysfunction in aging cells. Interestingly enough, the same is true of DNA repair. The major 4 families of enzymes involved in DNA repair also independently show these slightly slower rates of turnover. And so the rate at which you repair DNA errors go down, the mutation rate goes up exponentially with age. And it's not simply chronologic age, it's age of over the lifespan of the animal. So for example, it goes up exponentially in about 2 years in mice and about 80 years in humans. So the overall model then looks something like this. We have a number of upstream risk factors that we know about that have a huge impact on cell aging and cell dysfunction and a number of downstream outcomes. And regardless of what's happening, is that as cells divide, the telomeres shorten, they turn out to change the pattern of gene expression that slows molecular turnover. And as a result, you get dysfunctional molecules, dysfunctional cells, and age-related changes, such as those downstream. Now, that's interesting, but the question is, what can we do about it? And clinically, what we have tried to do for decades is to work on some of the upstream risk factors. For example, avoiding head trauma, preventing infections. For example, controlling hypertension and hyperglycemia by making patients euglycemic and lowering the vascular risks. We have a number of approaches we've taken, avoiding toxin exposure like paraquat and dioxin. And these have not been sufficiently effective. And more recently, in the past 2 decades, we've tried to intervene downstream, for example, with monoclonal antibodies to go after beta amyloid plaque, tau tangles, and a number of other approaches, but again, with insufficient efficacy. The question is, can we intervene effectively in cell aging? Well, it's already been done. 23 years ago, this paper came out in Science by Andrea Bodnar and her group out at Jern Corporation in Menlo Park. And what she showed is that you can take old human skin cells, in this case, and use telomerase to reset them. So morphologically, histologically, physiologically, and in terms of actual patterns of measured gene expression, they resemble young normal human cells. So we know we can reset aging in human cells. And in fact, we know we can do this as of 21 years ago in human tissues as well. This is a paper by Walter Funk in experimental cell research, again, the group out at Jern and Menlo Park. But what he showed is that you can actually regrow young human skin from old human skin cells if you use telomerase. So we can reset tissues. And in fact, you can do the same thing with human osteocytes, and you can do the same thing with vascular endothelial cells. You can essentially take old human skin cells or old human vascular endothelial cells from damaged coronary arteries and grow young arterial tissue. But can we do this with organisms? Well, the first paper that looked at that came out about 10 years ago from Ron DePino at Harvard. But he used an approach where he altered the germ cell lineage and got remarkable results. For example, regrowth of brain tissue and improvement in behavioral function and a number of other indices, biomarkers. But a better approach was that taken by a friend of mine, Maria Blasco, the director of CNO in Madrid. And what she did was use an AAV vector to use mouse TERT and reset the function of the brain cells. And not only do you see physiologic improvement throughout the rest of the body, but you see behavioral improvement. These animals normalize the behavior to that typical of young animals. So the question is, could we do this with human beings? Well, it's an interesting question. One I raised in the paper in the upper left, and a number of other people have begun to think this is an interesting idea. And in fact, what we're talking about then is using telomerase to reset cell aging, therefore resetting all the common biomarkers we talk about at this or other conferences, and as a result, reset clinical disease. There are a number of papers that bear on this. They're on the bottom, and all of those have active hyperlinks with the door reference, should you like to follow them up. The same is true when we look at common biomarkers, for example, beta amyloid, tau proteins, mitochondrial proteins, and so on. We see that telomerase loss or telomere loss is associated with slower turnover, and telomerase itself resets the gene expression and is protective. And again, references are there for you. Same is true of normal function. We find that not only do you increase the brain volume to much closer to young normal levels, but you get much better neural stem cell proliferation, differentiation, viability, and migration, and the telomerase decreases senescent phenotypes. So there's a lot to recommend this approach, Likewise, with regard to mitochondria, we know that telomerase improves the membrane potential, respiratory chain function, and also decreases tau protein pathology and reactive oxygen species. And again, references there for you. Same is true of inflammatory biomarkers. No matter which ones we've looked at so far, we find that telomerase tends to decrease the inflammatory changes. Safety is another issue. And what we find is that telomerase improves physiologic measures, and it's also well tolerated without adverse effects in animal studies. Moreover, it does not increase cancer risk. It is a much more complicated issue than most people realize. And the best review is the one that probably Kurt Whittemore, who worked on the animal data with Mary Bosco. He and I wrote a paper came out this year about what really goes on with regard to telomerase and cancer. In some respects, it's probably protective rather than risk. So the outcome is that we intend to take this to human trials. And while our emphasis will be on efficacy and safety, we're also very sure that we want to make sure that credibility stands up and that this is a rigorous test. And as a result of that concern about credibility, we've been able to attract a remarkable group of people on the scientific advisory board, including Dick Mose, who wrote that editorial with the vegetarians 6 years ago. Suzanne Hendricks, as many of you know, was involved in some of the design of the anakinumab study. Kurt Whittemore was a postdoc who worked on the animal paper with Mary Bosco. Joe Araujo does large animal Alzheimer's work out of Toronto. And Mamouna Zuz actually runs translational gene therapy work on neurologic disease in the UK. The same is true of our clinical advisory board. We were very specific and we wanted to attract not only people who had a global reputation for integrity, but who were skeptics. So in all cases, we wanted people who were not convinced this model was true, but they found the model intriguing and were with us and wanted to make sure we test it rigorously, which we intend to do. What we're going to be using is a single dose AAV gene therapy with human telomerase gene in a customized cassette and addressing this question. Is it possible to cure age-related dementias such as Alzheimer's disease? And the answer is, we'll see. It very much, like everything else, depends on the data. Good theory is one thing, good data is another. So thank you very much for joining us. I hope you found it an intriguing presentation. For those of you who would like to talk to me further, please do, and I hope you'll enjoy the remainder of the conference. Thank you.